Hello everyone, my name's Stefan. I'm a first-year master's student at the University of Ottawa, working in the MER lab. I'll be going over my research on how isoform level analysis can improve prognostic modeling in acute myeloid leukemia, or AML. Accurate prognosis is important in cancer because it helps guide treatment decisions and directly impacts patient outcomes. One common tool for this is gene expression profiling, which tells us how much different genes are being expressed in a tumor at a specific point in time. But genes often produce multiple mRNA isoforms through a process called alternative splicing, and gene-level data simply sums all of these isoforms together. That aggregation can mask important differences, especially when those isoforms have distinct or even opposing roles in cancer. This complexity is especially relevant in AML, which is a highly aggressive and heterogeneous blood cancer. And recent studies suggest that important biological signals might be hiding at the isoform level, completely missed when we only look at the gene level. So the goal of this project was to see whether isoform-specific expression could provide additional insights into AML prognosis, beyond what we can get from traditional gene-level analysis. To explore this, we analyzed isoform-level expression and clinical data from 4 AML cohorts. We processed the RNA-seq data, quantified transcript-level expression, normalized it, filtered out low-variance isoforms, and z-scored the data. We also gathered clinical variables, which we used in multivariate models. To assess prognostic value, we ran Cox proportional hazards regression for each isoform. We then combined results across the 4 datasets using random effects meta-analysis. Lastly, we performed pathway enrichment analysis to determine whether particular biological processes were enriched among significant isoforms. We started by running univariate Cox regression on isoform level expression in each of the 4 AML cohorts. Figure 1 shows volcano plots of the results for each dataset. Each point is an isoform. Those to the right are associated with higher risk of death, and those to the left are with better survival. The y-axis shows statistical significance. Across all 4 datasets, we found hundreds of isoforms significantly linked to patient survival. Interestingly, the specific isoforms varied between cohorts, which points to both biological variation and dataset-specific differences. To account for variability between datasets, we performed a meta-analysis, combining the Cox regression results from all 4 cohorts. Figure 2a shows a volcano plot from the meta-analysis. We identified many isoforms that were consistently associated with survival across datasets. One example is shown in 2b, NPM1-210, which is an isoform of the well-known AML-associated gene NPM1. Interestingly, this isoform wasn't significant in any single dataset, but became significant when we combined the results. And this shows the power of meta-analysis. It helps uncover subtle but reproducible associations that might otherwise be overlooked. Next, we asked whether these isoform-level associations held up after adjusting for clinical variables like age, sex, white blood cell count, and karyotype complexity. Figure 3a shows a volcano plot from a meta-analysis of multivariate Cox regression results. Even after adjustment, several isoforms remain significantly associated with survival. In Figure 3b, we highlighted one example, MAS2-201. This isoform was significantly associated with poor survival, even after adjustment for clinical covariates. While MAS2 has been linked to other blood cancers, its role in AML hasn't been reported before, suggesting that MAS2-201 could be a novel biomarker. Up to this point, we focused on isoform level expression. In Figure 4, we directly compare this to gene level results to ask what might we miss by only looking at genes. Figure 4a shows a Sankey plot comparing significance at both levels. We found 875 genes that weren't significant overall, but had at least one isoform that was. These are cases where the gene level view completely misses transcript-specific associations. In 4b, we rank these genes by the percentage of significant isoforms. One of the top hits is FLI1, a transcription factor involved in hematopoiesis and implicated in some AML cases. Figure 4c shows that while FLI1 as a gene was insignificant, isoforms like FLI1 203 and 204 were strongly associated with poor survival. This is reflected in D. Kaplan-Meier plots show no clear difference at the gene level, but a strong survival difference for FLI1 204. So this reinforces the main idea of our study. Isoform level data reveals important patterns that are lost at the gene level. To understand the biology behind these isoform level findings, we ran pathway enrichment analysis using hazard ratios from the Cox regression. In both 5A and 5B, we selected the top 10 AML-related pathways that were significantly enriched at the isoform level, but not at the gene level. Figure 5A shows results from gene set enrichment analysis, and 5B shows overrepresentation analysis. Among the enriched pathways were cell cycle, apoptosis, and DNA replication. Importantly, we also saw enrichment of the acute myeloid leukemia and chronic myeloid leukemia pathways, highlighting just how clinically relevant these isoform level signals are. These are well-known AML pathways that were completely missed in the gene level analysis. So to summarize, first, isoform level analysis revealed survival associations in AML that were missed at the gene level. Second, we found that many enriched pathways, including those directly related to AML, only emerged when we looked at transcript level data. Altogether, this work shows that alternative splicing carries important prognostic information and that integrating isoform level data could lead to more accurate models and better biomarkers for AML. This work was supported by the Leukemia and Lymphoma Society of Canada and NSERC. I'd also like to thank the MER Lab for their guidance throughout this project. Thanks for listening.